All right. So today is Unit 3, Day 1. Today is the start of our third unit. I will give you all, I'm going to cut this down a little bit because the warm-up is pretty short, but I'll give you all about 5 minutes to knock out the warm-up, and then we'll jump directly into the lesson. And I'll talk a little bit more about some other stuff as well.
Is it okay if we don't do good on it?
Yes, but you should be able to do pretty well on it. It's basically review.
Oh boy.
Okay, great. So, this is the first slide. I should have another slide. We are starting our third unit on cellular reproduction today. This will be a 7-day unit, which means that we'll have 5 days this week, and then we'll have 2 days next week. So the last day of our third unit will be next Tuesday, September 22nd. So again, we're moving at a pretty rapid pace here, and I hope you all are able to really grasp these concepts at the speed we're moving. But if not, you also always have the opportunity to reach out to me and say, hey, Mr. Rudd, this is moving too quickly. I've had a couple of those conversations with a couple of you over the last few weeks when a few people have reached out to me and said, Mr. Rudd, I need you to slow down a little bit, or I need some more one-on-one time. And I really do commend those people for doing that. So you all should always be in the habit of reaching out to your teachers and saying, I need you to do this a little bit more or a little bit less or a little bit differently, because ultimately we are serving you. This is your education. You are getting from it whatever we can give you. And so you need to be in a position to say, hey, this is not what I need. I need you to do it this way. And you do it in a respectful way, obviously, but at the end of the day, we're serving you. And so if the product needs to be changed a little bit, then we do rely on you all to do that. But anyway, we will be starting our third unit today, and it'll go until next Tuesday, and you'll have one assignment a day. You'll have a warm-up, one assignment, and an exit ticket. So I'm hearing from some of my former students who are not in biology, but in other classes, they're saying that they're getting a lot of work from their teachers. And so we're trying to make sure that we're not overloading you all or overwhelming you all with the amount of work that we're giving you. And so you will only have one assignment a day, and it totally can be done in the 35 minutes that we have for the asynchronous portion of class. Today's lesson might run a little bit long, so I'm going to jump into it, but you'll still have plenty of time to knock out the assignment and the exit ticket at the end of the day. So as we start this third unit, we have to remember that biology is a cumulative subject, which means that each successive unit is building upon what we've learned prior to that unit. So units 0, 1, and 2 are still coming into play and still foundationally important as we start unit 3. Unit 0, we introduced the concept of Sternger. Sternger was an acronym that allowed us to explain the 8 essential life processes. And so we're going to continue to touch back on Sternger, but in this specific unit, we're talking about one of the R's. We're talking about reproduction. And what you all will see this week and going forward is that I'm going to have some translations. I'm translating most of the text on these slides into Spanish, just to be a bit more inclusive, and also in recognition of the fact that tomorrow is the beginning of Hispanic Heritage Month, National Hispanic Heritage Month, in recognition of all of the contributions of the Hispanic community, which is really a community of many, many different ethnicities and nationalities, but specifically just in general of the Hispanic population in the United States, the contributions that they've made to our culture, our politics, our scientific advancements. And we'll talk about some of those things as the Hispanic Heritage Month begins tomorrow and runs through October 15th. But I'm going to do this because there are some students who benefit from the translation. And if there's another language that you all would like me to translate into, then we can discuss that as well. So get used to saying some Spanish on these slides. But the R that we're talking about today is, and for this entire unit, is reproduction. Reproduction is the production of offspring by a sexual or asexual process. So there is a difference between sexual and asexual, which we will talk about going forward. But reproduction is used to make new cells, and it's also used to copy and preserve DNA. Most cells will reproduce in their lifetime. Not all cells, but 99% of cells will reproduce in their lifetime. Does anybody know, can anybody provide an example of a cell that does reproduce?
Which way, though?
It doesn't, well, we'll talk about it. That's a good question. That is a good question. That's an important distinction. But for right now, it doesn't matter. Your skin cells, for example, your skin cells reproduce constantly. If you have a slight scratch or a cut that breaks the skin, then some of your skin cells will die, but they will swiftly be replaced by new and healthy and living skin cells. So that's a good example. The cells in your liver. Your liver is one of the few organs in our body that can essentially regenerate. So when people have different cancers or different diseases of the liver that require surgical removal of parts of their liver can literally regenerate and return to its original size, typically in about 6 months, maybe even less than that, depending on the amount of tissue that was removed. So the liver is a really dynamic and regenerative organ. But the cells that are in our nervous system, so our neurons, the cells that are in our brain, and the cells that make up our spinal cord and most of the nerves throughout our body, do not regenerate very well, which is why people who are paralyzed essentially remain that way for the duration of their life. And sometimes this is not the case. Sometimes people get, like, let's say you have a football injury and someone is temporarily paralyzed, they can eventually recover some of the mobility in their body. And that's largely because the cells have not died. They've just been stunned, and they've basically been put on pause while the body kind of recuperates itself. But once those nerve cells die, they're not replaced. And so this is why people who are addicted to drugs really put themselves at great risk of neurological damage, because once those nerve cells have been damaged, there's really no way of replacing them. We are making scientific advancements in what's called stem cells, and we'll talk about that later in this unit and throughout the course, but nerve cells do not reproduce very well. Okay, so what you'll see is that the things that I think you all should write down in your notes are highlighted. The English portions are highlighted in yellow, and the Spanish portions are highlighted in green. So when we talk about cellular reproduction, essentially what we're talking about is cell division. And cell division.
Can you say that again?
Yes. So when we talk about cells reproducing, what can I say? A synonym of that is cell division. So those two terms are essentially interchangeable. Cell reproduction and cell division are basically the same thing. Cell division is the process by which individual cells and unicellular organisms reproduce. Now, when we're talking about multicellular organisms like you and I, or the plants that you can see outside, they reproduce via different methods, which we'll talk about later this week. But cell division is the simple process that allows one cell to reproduce, one cell to essentially copy itself. Okay. And here are some images that really depict cell division pretty well. We've got a plant cell on the left. You can literally see the orange stuff is the genetic material. Those are chromosomes that are literally being pulled apart. And so we're starting off in one cell, and then they're pulled apart, and you can actually see one cell being split into two. So the cell membrane splits, the cytoplasm splits, and now we end up with two cells. The image on the right is a colony of bacteria, and you can see just how quickly cells cell populations can grow. So one cell becomes two cells become four cells become eight becomes 16, 32, 64, 128, 128, 256, 256, so on and so forth. You get the point. All right, so this is how colonies of bacteria are able to grow so rapidly. This is not a linear growth. It's not 1 to 2 to 3 to 4 to 5 to 6. It is a exponential growth. Okay, and this is what is essentially happening in our bodies, but our cells have the ability to control themselves under most circumstances and say, we don't need any more. We don't need to continue to reproduce. So we're going to put things on pause for now. When that does not happen, you end up with cancer, but we'll talk about that later in the lesson. All right, so our bodies, like I said, are able to rebuild themselves. And in the course of a year, many of the cells in your body will be replaced. Not all, but many. Some things replace themselves very quickly, and some things take much longer to replace themselves.
Do we draw this?
You do not need to draw that. But I just want you to answer the question. Why do we think new cells need to be created?
Can I
Yes.
guess? I think why new cells may need to be created because old cells may die off, shrink, something like that.
Excellent. Yeah. So old cells die. That's one of the primary reasons why we need to create new cells. The lifespan of most cells is not very long. What else? What's another reason? Nobody else?
To like grow.
Excellent. Yeah, that's another primary reason. So organisms grow. You don't go through life the exact same size that you were born. So we have some need to grow in size and also to become more organized as we go. So we need new cells for that. We'll talk about, there's a third reason as well that we'll see in a couple of slides, but really quickly, as a flashback, what do you remember from previous units? Which of the 4 macromolecules is DNA?
Nucleic acid.
Excellent. DNA is a nucleic acid. And so just to push you a little bit further, what is the monomer of DNA in that case?
Nucleotides.
Nucleotides. Good. So DNA is a nucleic acid, which means that it is made up of nucleotides, because nucleotides are the monomers of nucleic acids. And then I'm going to push you even further. Big brownie points to whoever can get this. What are the 3 parts of a nucleotide? I love that I can see Gabby actually going back and looking in her notes. At least that's what I assume she's doing. She could be playing Animal Farm or something on her phone. Animal Crossing. I said Animal Farm.
Phosphate, nitrogen base,
okay. Good.
And the sugar.
Excellent, Castro. Thank you so much. So we've got those 3 parts. We've got a phosphate group, we've got a nitrogen base, and we've got a 5-carbon sugar. So those are the 3 parts of a nucleotide. So I love that we're able to do that. And I can tell that people are looking back at their notes and they're really recalling information from the past. That's a great way to be a great student. And that's what's required really to be a good student in this class, because we have so much information. But DNA is an example of a nucleic acid, which is one of our 4 macromolecules. Nucleic acids are polymers that are made up of nucleotides, which are monomers. And nucleotides are made up of three things: a nitrogen base, a phosphate group, and a 5-carbon sugar. So that's how we can wrap it all up. And those are the connections you all need to continue to make mentally as we continue to introduce more concepts. Excellent job. All right, so again, more TBT, but the following terms are synonymous with DNA and genetic information, meaning they can be used interchangeably, really. But there are some important differences between them. We've got chromatin, sister chromatids, chromosomes, homologous chromosomes, and genes. That sounds like a lot, I know, so let's just talk about it a little bit.
Did we need to write that down? Because I was in the middle of writing all that stuff down. I was going to draw a picture of it too.
You don't need to draw pictures. And like I said, the stuff that I really want you to write down will be highlighted, but I'll do a better job of pointing it out to you. But no, you do not need to draw pictures unless, you can draw something really quickly and it's going to be helpful to you. All right. So chromatin over here on your left, and this might be a good idea to, something that you should write down. In fact, I'm going to highlight it just to make it easier for you. Okay, so chromatin is a picture on the left. It looks like a big bowl of spaghetti. So this is actually inside of the nucleus. Chromatin is random and disorganized DNA. We can see there's basically no order to this. It's just twisted and
Why is it like that?
Why is it like this?
Yeah.
That's just how it's stored in the DNA. I'm sorry, in the nucleus.
So it's just like that.
Yeah, it just looks like, and the reason there are some physics involved and there's some chemistry involved about why exactly, but essentially this is the least energetically taxing way of storing the DNA. So it requires the least amount of energy to do it this way. And that's kind of what you'll see. That's one of the themes in science in general, is that nature is really good at doing things in the most efficient way. We don't want to use more energy than we need to. So just basically allowing the DNA to just be there in the nucleus in a totally random and unorganized way requires very little energy. But then as we begin to organize it into chromosomes, that takes some energy to actually wrap it up very tightly. So the chromosomes are condensed and they're organized. Chromatin is random and disorganized. And these terms are they're similar. They take a long time to really differentiate between. I'll be honest, even after my third biology class, so I took high school biology, then I took AP biology, then I took biology in college. Even then, I was like, okay, wait, which one is which? So these concepts, these terms, specifically because they sound alike, we've got chromatin, chromatids, chromosomes, they sound alike. So it takes some practice to get to know which is which. But chromatin is like the big bowl of spaghetti. Chromosomes are more organized. They look like the letter X. And I'll move on. So we can break down chromosomes even further. It might be a good idea for you to just draw a simple X in your notes. To represent a chromosome. And a chromosome has specific parts. The ends of the chromosome are called telomeres. Telomeres in Latin essentially means end part. And those telomeres are there as protection. So they prevent the chromosome from deteriorating or from fusing to another chromosome that might be next to it. So they're pretty important. We've got sister chromatids. So if we have an X, then we can imagine that one half of the X is one chromatid, and the other half of the X is another chromatid, and together they are called sister chromatids. But they are identical copies of DNA. In theory, they should be identical copies.
Do we write this down?
I would just I would draw an X and then I would just highlight, telomeres, draw an arrow to telomeres, centromere, draw an arrow to the centromere, and then a brief definition of sister chromatids.
Okay.
Before you go to the next slide, I want to let you know I'm still writing.
Thank you. So the centromere is just the region where the two sister chromatids meet, where they're connected. Okay. All right, so let's zoom in a little bit. We know what a cell looks like at this point. We dedicated Unit 2 to talking about the cell. And we know that inside of the cell, inside of a eukaryotic cell, we have a nucleus. And that is where the genetic material is stored. So I'd write that down. Step 1, the genetic material is stored in a nucleus. The cell's genetic material is stored in a nucleus. But we can zoom in even further. And within the nucleus, there are chromosomes. The genetic material in the nucleus is organized in chromosomes. That's step 2. And in step 3, we're zooming in even further. Chromosome.
Mr. Rudd, you're
Okay.
moving too fast.
Of course, you don't need to write down. You're welcome. You don't need to write down both languages, just the one that you are most comfortable with. And then step 3, chromosomes are tightly wound DNA. Humans have 46. And other organisms have more or less. In fact, the number of chromosomes does not correspond to the sophistication of the organism. What I mean by that is humans have 46, but fruit flies have like 98. And of course, fruit flies are much more simple organisms than humans. So the reasons for that are still somewhat unknown to us.
Mr. Rudd,
Yes, ma'am.
you'll never believe what we saw. I saw a flying roach. Not in the house, of course, but somewhere where we were going on walking on the trail. I saw a flying roach.
Yeah, I've seen a couple of those in West Mech.
Really?
Yeah, the oldest apparently the oldest roaches have wings, and that was something that I did not know until I saw one, and I was like, "Is that roach flying?" But yeah. Okay, you don't need to write anything on this slide down or draw anything. This is just here so I can talk. So we're going to talk about the cell cycle, and the cell cycle is essentially the cycle through which cells reproduce. And keep in mind, when we say reproduce in reference to a cell, we're talking about the cell dividing. So in this case, you can see these are two cells that are essentially growing out of what was one cell. Their genetic material has been split. Their nuclear membranes are separating. And this is happening almost constantly throughout your body for most cells. Like I said, your nerve cells don't really do this. Now, I've highlighted things strategically here, so please write down what is highlighted in your preferred language. But in general, there are two cell types. The first cell type is called a gamete, and these are sex cells. And then there are also somatic cells. Your sex cells are sperm and eggs. Your somatic cells are all cells besides those two sex cells. Okay, so nerve cells, muscle cells, digestive cells, skeletal cells, these are all somatic cells. And somatic cells, the word somatic means body. Okay, so when we see the prefix soma, S-O-M-A, that means body. And so that can kind of help you keep in mind that somatic cells are throughout your body. They're everything except for the two types of sex cells. I'll give you about 10 more seconds to write. Okay. All right, so why do we need to make new cells? Well, I can't remember if it was, well, I think this one was Ayana. Ayana said that we need to make new cells because old cells die. So really, all you need to write here is just replacement. Why are new cells made? One, replacement. Shouldn't take too long. Old cells die and must be replaced. Reason number two why new cells are made, growth. This was from Gabby. Gabby reminded us that organisms grow in size over time. So, in fact, in the first 9 months, well, not nine, well, while you are in utero, so while humans are inside of their mothers, we grow very, very rapidly. And so, actually, as we get older, our growth slows down a little bit. So, the fastest growth happens earliest in life, but we continue to grow. In fact, most of you probably still have several years of growth.
Do adults grow, even if a little bit, or you just stop growing?
Well, you stop growing, but there are still important changes that happen in an adult's body. There are specific organs that grow larger and some that grow smaller. There are specific parts of the brain that grow more complicated and more sophisticated, and there are other parts of the brain that obviously become less functional as you grow older. So there are still important biological changes that happen. And in fact, there are specific fields of science and medicine that focus on different stages of life. So many of you probably have attended at some point a doctor's appointment with a pediatrician who is a doctor who focuses solely on children, the health of children. Then there are gerontologists who focus solely on the health of elderly people, but then there are also doctors who focus on people in the middle stages of life. So each stage comes with its own challenges and its own unique benefits. Good question. And then our third reason why cells are made are for reproduction. And this can be sexual and asexual, but we need to be able to produce new offspring. Okay. So we've got those three reasons. And this is essentially what it looks like. So you can see the orange genetic material. The green stuff are called spindle fibers, and they pull the genetic material apart. So we're starting off with one cell, and we're ending up with two. You may be wondering how we're able to get these images. We can use specific dyes that are attracted to specific chemicals. So likely there's a dye that is being used that is attracted to the nitrogen in DNA. And so it turns the DNA an orange color, which makes it easier to see. And then the green stuff, like I said, it's called the fiber. It's a specific kind of fiber. And so it's probably very heavy in protein. So we might be using a phosphorus-based dye for that to turn it green. And so that's what gives us these nice, pretty easy to see images. Okay. The cell cycle is the process cells go through to make new cells. Okay, that's really all you need to write on this slide. But I'll talk about the other parts as well. The cell cycle is made up of different stages. The first stage is called interphase. And interphase is also made up of 3 separate stages. And we're going to talk about them in the coming slides, so you don't need to write this down right now. After interphase, we undergo what's called an M phase. So M phase can either be mitosis or meiosis. Again, we'll talk about it in another slide. And then the last step would be cytokinesis, which is when the 2 cells actually split apart from one another. But in this case, this pie chart allows us to really see that interphase, which is G1, S, and G2, is by far the longest stage of the cell cycle. It takes up about 90% of the cell cycle, and then the rest is M phase, and then the cell splits. Okay, but the cell cycle is the process cells go through to make new cells. Here, again, this is where you can write it down. There are 3 major parts of the cell cycle. The first part is interphase. The second part is M phase. And the third part is cytokinesis. So I'll give you all some time to write that. Okay, does anyone need more time here?
No.
Good. Okay. So again, we're focusing on what is highlighted. So within interphase, which is the first step, interphase is the longest phase of the cell cycle. And the cell is preparing itself to divide. And what happens is that there is a G1 step in which the cell grows. So G stands for growth, okay? Well, it kind of stands for gap, but I've always liked to think of it standing for growth. Then there is synthesis. Synthesis is the most important part of interphase. Synthesis is where the DNA is replicating. And then there is a G2 step, which is, again, more cell growth. Please do write down that synthesis is where the DNA, or is when the DNA replicates. Okay. Okay, so just a little bit more about DNA replication. During replication, the DNA is copied. Okay, so we can see what happens. This is one strand of DNA. We can see the double helix. That double helix gets unwound, and each strand is getting copied. And we're moving up the strand. We're moving up the strand, up the strand, up the strand, and eventually you end up with two distinct strands of DNA. They're copies of each other, though. So we're making two strands from one strand.
Does it actually look like that inside of the body, or no?
Essentially, yes. There's a lot more going on. There would be there's a specific enzyme that is needed to unwound the DNA called helicase, and it looks like a zipper. There are specific enzymes that are required. There are specific proteins called small binding subunits or something like that that are required to make sure the DNA doesn't fall apart. There are specific enzymes that are required to actually do the copying called RNase primers. So there's a lot going on, but in short, yes, it does look like this. There'll just be a lot more things along the strand. And that's above the level of this course. So all you need to know for now is that during replication, the DNA is copied. Okay, there's a lot here, so I'll spend some time here. The second step of the cell cycle is M phase. And during M phase, the chromosomes in the nucleus divide. And there are two types of cell division. So M phase can either be mitosis or it can be meiosis. Mitosis is used for asexual reproduction. And mitosis gives you an exact copy of the original cell. They are genetically the same. So somatic cells, like the cells in your liver or your bone cells or your muscle cells, somatic cells undergo mitosis, as well as unicellular organisms like bacteria. But in order to produce sperm and egg, which are your gametes, we undergo meiosis, which is asexual reproduction. And there's a way I like to think about this. I wish I had put it on the slide, but mitosis has that IT. It's MIT. So when I first learned about this in high school, my teacher told me that the IT, we can think of it as being identical twins. So we end up with two copies of the same cell. They're identical. And then meiosis has an E and an S. So the E stands for egg and the S stands for sperm. So meiosis gives us our sex cells. Okay. So we can kind of think of it like this. This is a good image to represent the stages of mitosis. You start off with one cell, then that cell begins to divide and it begins to move apart, but it's not totally distinct. We don't have two totally distinct cells yet. And then eventually we get to two totally distinct cells, which are separate from one another. They have their own nuclear membranes and their own cell membranes. And then the last step of the cell cycle is cytokinesis. Please do write down everything that is highlighted. I think I messed up my colors here, didn't I? Yeah. Cytokinesis means quite literally cell movement. Cyto means cell, kinesis means movement. So cytokinesis means cell movement. And this is when the cytoplasm and cell membrane of the parent cell split into two cells, which we call the daughter cells.
So, how long is this unit again?
7 days.
Wow, that's a lot of notes.
Okay. So cytokinesis is when the two cells literally move apart, they split.
Mr.
Yes, sir.
I missed the slide before the phone because my dog was doing something.
Okay. This one?
Yes.
Can you take a picture of it?
Yes, I can.
Okay.
All right, I'll take a picture.
Okay, thank you. All right. So we can see this happening in slow motion in this animation. The cell is being cleaved, which means it's just being basically cut apart. And then cytokinesis allows the cells to actually move. So there are what are called motor fibers that allow them to actually move apart. And we can see this in a video in the future. So again, putting it all together here, the 3 steps. This is a very brief summary that allows you to see, what is happening during the cell cycle. We have interphase, which again is the longest step, the longest phase of the cell cycle. And interphase is G1, synthesis, and G2. And then we have M phase, which could be mitosis or meiosis. And then cytokinesis, when the cell splits. Now, one thing that we should note here is that there are different checkpoints represented by these red bars. They're checkpoints along the cell cycle. And those checkpoints are there to prevent this cancer. So all you need to write down here is the definition of cancer.
Is that an ear or what is that? In the
that is actually a colon. So that is someone's large intestine.
That's what it really looks like.
Yep. Okay, so cancer is go ahead, question.
How did they like get the picture like that, like inside of somebody?
Do you really want to know? Essentially, they insert a small camera, and it is on a long tube of sorts. So just imagine you can see this cord that I have here. At the end of this cord, there would be a camera, and we insert it not through the mouth, we just not through the mouth. No, not through the mouth. If we were looking at the stomach or if we were looking at the esophagus, then obviously the mouth would be the easier entryway. But your large intestine is the very last part of your bowel before we get to the anus. So it makes more sense to go in through the back door, essentially.
Yeah. You gotta explain it.
Yes, but there'll be a camera. It has a flashlight on it. And this is actually a pretty common procedure when people are having intestinal issues. rather than doing a surgery, this allows us to actually see what's going on instead of having to just cut inside of someone.
Now, what's the picture on the right? Are those cancer cells? I meant on the left.
Yeah, those are cancer cells that are dividing rapidly and they're not being regulated. So cancer is an unregulated growth of abnormal cells. These cells typically are mutated in some way, and their growth is not being controlled like it should be. Most cells have a programmed lifespan, and so within the DNA of the cell, they know, okay, once I hit 14 days old, then I'm going to die, and that's just the natural way of things. That's healthy. And so that's called apoptosis. Again, that's above the level of this class, but cells have a timeline that they should be on, and they should know when their time is up. But cancer cells don't know when their time is up. They just continue to reproduce. Essentially, they'll continue to reproduce unchecked unless some sort of therapy is put into place, whether that is surgery or chemotherapy or radiation. Something needs to be done to stop their growth.
So
Why don't they know when to stop? I mean, when their time is up?
That typically happens because of a mutation. So within the DNA of the cell, something has gone wrong. That means that there's no regulation. The cell is not able to tell itself, okay, I've reached this point in my life, so I need to reproduce. And then when I reach this point in my life, then I need to die. Something is not telling that cell to die. And so it just continues to live and it continues to reproduce. And that is a genetic issue. So sometimes you'll hear, as science has advanced, we've been able to analyze people's DNA, and we can say, like, your cells, let's say the cells in your breast tissue are specifically predisposed to developing cancer because they don't have that checkpoint to say, like, hey, your lifespan is up. It's time for you to go ahead and die. So we can actually tell people that now before they develop cancer, like, hey, you are actually at greater risk of developing cancer because your cells don't have that checkpoint. So it's good that we know it, but it also means that, in some cases, cancer can develop.
I have a question.
Yes.
Do the cancer cells stick to healthy cells, or do they just spread all over your body? How does it work?
Cancer can spread, and that's a process called metastasis. So sometimes you'll hear that some people have metastatic cancer, which means that the cancer has spread to other parts of their body. So in this case, let's say this person has, stage 2 colon cancer. The cancer cells in this tumor can spread through the blood to other parts of the body, and it can lead to lung cancer as well. And so that's really one of the reasons why some cancer treatments are so aggressive, because the doctor will want to stop the potential spread of the cancer to other parts of the body. So you need to go ahead and move the cancer. You need to kill the cancer cells as quickly as possible before they have an opportunity to move to other parts of the body. And typically when people are in stage 4, that means that the cancer has spread to other parts of their body. Okay, so we can start off as literally just 2 or 4 or 8 cancerous cells can very, very quickly become hundreds of thousands or millions of cells that lead to a tumor. And that's what makes cancer cells so problematic. But this is what I was talking about. In a healthy cell, there are specific checkpoints that regulate the cell cycle. And you should write this down. These checkpoints prevent potential mistakes, not only mistakes that can lead to cancer, but mistakes that could just lead to dysfunction. So, for example, there are specific conditions in which a person's stomach cells no longer produce the mucus that is needed to line the stomach. And so that can mean that you end up getting, what are they called? Essentially, you end up getting wounds in your stomach because the lining is not there. So these checkpoints are there to say, like, hey, at this point in your life, you should be doing this. At this point in your life, you should be producing this protein. At this point in your life, you should reproduce. And at this point in your life, you should die. If those checkpoints are there or not there, we can end up with cancer or other harmful conditions.
Can you be healthy and still have cancer?
What's that? You broke up a little bit, Ayana. Can you say that again?
I said, can you be completely healthy and still develop cancer?
Yeah, you can. You know, there are some people who live potentially years without knowing that they have cancer. And that's one of the most dangerous parts of it, because some people don't realize that they are harboring this tumor until it's basically too late for them to do anything about it. So you know, healthy people can get cancer because some of the cancer that we can develop is totally genetic. You can have, you can follow a perfect diet, you can follow a strict workout regimen, but if the DNA inside of your cells is damaged in some way, or just abnormal in some way, then you can still develop cancer.
Are cigarettes
Here
more
now.
This is what makes cancer so difficult to cure, because if it were always just a matter of, oh, you just need to stop eating hamburgers, then, you know, it will be a much easier problem to deal with. But there are so many different causes of cancer. And Adrian, I heard you mention one cause.
Well, I was gonna ask, are cigarettes more likely to cause cancer than dip or chewing tobacco?
Cigarettes are more likely to cause lung cancer than chewing tobacco, but chewing tobacco is more likely to cause oral or tongue cancer than cigarettes. So,
yeah,
you
but
know,
it's still pretty low, right?
what, dip?
Yeah. Like, as long as you, like, you know, brush your teeth every day.
If you are, you know, the chances kind of accelerate the more you do it. So, you know, if you've only, even with alcohol, right? So if you only drink once or twice a month, or if you only do dip or chew tobacco once or twice a month, then your chances will remain pretty low, because your body has specific defenses against those, you know, poisons essentially. But if you're doing it, you know, every day, very quickly your chances of developing these cancers increase, because our bodies just are not capable of eliminating those toxins from the body, eliminating the alcohol or eliminating the nicotine. So most things in life are good, are okay in what am I trying to say? Not moderation. Thank you. Moderation. That doesn't, I'm not advocating for the use of anything, you know, you all are still young, so I'm not advocating for that, but our bodies are very, very resilient, and our bodies can handle things in moderation. But when you start to flood the system, that's when you will run into problems. All right. So another image, I'm not going to focus on this really, but there are some, you don't need to write this down. Here are some questions that your cells would ask itself at these different checkpoints. Are my chromosomes ready for separation? Is the environment okay? So before I actually divide, you know, do I have enough room to divide? Do I have the right nutrients available to divide? Is my DNA intact? So cells should not divide if the DNA has been damaged, because again, that can lead to cancers. Is my DNA correctly replicated? So have I copied my DNA correctly before I move on? So these are just some general questions that need to be asked before the cell actually does the process of dividing. All right, so that lesson was really long. We only have 5 minutes left in class. You can still do the exit ticket. This practice is also very, very short. I think it'll probably take you all 10 minutes. So we can finish this before third block. But I apologize for taking up so much of the time today. Today's lesson was just long.
Is the next 7 days notes like this too, as well?
Today's lesson is the longest.
That's good.
I want to ask something about like the cells and like the nervous cells.
Mm-hmm.
So basically, when somebody of old age dies, does that mean all their like it's because all their brain cells die out?
Oh, you mean like of natural causes when people say like they died of old age?
Yeah.
Uh,
Or like when they start getting problems like Alzheimer's and dementia.
Okay, so those problems are specifically problems of the brain. So Alzheimer's, for example, is caused by a specific protein that prevents your brain cells from communicating normally like they would have. And in most cases, Alzheimer's starts in a specific area of the brain that affects memory. But then as time goes on, it will spread, that protein will begin to spread throughout the brain to affect other areas that have other functions like controlling breathing. So people who die of Alzheimer's can actually die of, essentially suffocation or asphyxiation. And so those are different than people who are just dying of natural causes, dying of natural causes or dying of old age. Typically, when that happens, it is a cause of, it's typically a heart problem. And it can mean that the blood is not pumping oxygen to the brain efficiently. And so, I think oftentimes when that happens, we don't have a real reason because there's no autopsy that is performed to actually say this person died because this happened. And so, we could oftentimes we could find an actual cause for dying of old age, but when you're 85, 90, 95 years old, the family may not be interested in, an autopsy being performed, especially if they died comfortably, in their own home and everything.
Mr. Red,
Yes.
the exit ticket, the third question, does G1 mean like cell growth or like G2?
I'm pulling it up now.
Like, it has G1 and G2 as options. Does that mean cell growth?
Yeah, so that's what happens in G1 and G2. So really what the question is looking for is for you to just put them in order. So what happens first? So what is step 1? Then what would be step 2, step 3?
Okay.
Am I looking at the right thing? Yeah, all you need to do is put them in order. Alicia, did you have another question? I thought I saw you come off of mute.
No.
Okay. But the cool thing about nerve cells, like I said, is they don't divide, they don't reproduce often. But this is where we're getting this new scientific field of stem cell research. And so we're going to talk about stem cells in a few days, but essentially stem cells allow us, well, in some cases, they allow us to replace those dead or damaged nerve cells. So this is how we're getting, some people who are paralyzed are able to recover some of their lost function. Okay, and with that, it's 11:35. So again, I apologize that I took up so much time today. This is the longest lesson of this unit. The actual assignment for the day, I think, won't take you all very long, so you can go ahead and knock that out in the next 10 minutes before a third block. Or, oh, actually, we have lunch, so you guys do have time to do it if you want to, but it's pretty short. So go ahead and knock it out, but I will apologize, and I'll talk to you all tomorrow.